Mostly today we're going to be talking about genetics, but I wanted to start off today talking a little bit about graphing. On every AP exam, there's going to be graphing. You've got to make sure you know how to graph. Typically, the kinds of graphs you're going to be asked to make are either line graphs or bar graphs, what we might call histograms. In some cases, they won't tell you which type of graph to draw. I will tell you that you want to do a bar chart when you're dealing with categories of data, and typically you're counting things in those categories. That's when you want to do a bar graph or histogram. You want to use a line graph when you're doing what's called continuous data. By continuous data, we mean like numerical kinds of data, not categorical. Here's the thing though. If the question doesn't tell you which type of graph to draw, I would say don't chance it and do what's called a scatter plot, which is what you see here on the screen now. So in a scatter plot, you plot your points just like you would on a line graph. You don't draw in the curve like you would normally do. You just leave the points. You also, on these graphs, can do error bars. And typically the kinds of error bars that we do is we go up from the point 2 standard errors of the mean and down 2 standard errors of the mean. You can see that all these points have error bars on them. So what I'm giving you advice to do here is if the question doesn't tell you line graph or bar graph, and you're not sure which one's appropriate, instead, just do a scatter plot. And the scatter plot essentially meets all the requirements of a line graph. It also meets all the requirements of the bar chart. So that way you're covering your bases if you do that. Something else I want to talk about is what goes where in your graph. Remember that your independent variable always goes on the x-axis. That's the thing that you're intentionally manipulating during your experiment. You're changing it to see what effect it has on other things. That's your independent variable. It always goes on the x-axis. Your dependent variable, on the other hand, the thing that changes because you changed this thing, goes on the y-axis. Another couple of pieces of advice to make sure that you know, they're going to give you an axis on the test. So you've got to use the axis provided. Another important thing is that your whole graph has to fit on that axis. So you've got to plan in advance before you start graphing. And when I say your whole graph, I also mean the error bars. Your error bars can't extend up here above the graph like that. They have to fit on the axis. So plan your graph beforehand, make sure it's going to fit. Also, I will tell you that even though you're supposed to do your FRQ answers in pen, whenever you're doing graphs, I'm going to tell you to graph in pencil. And then once you're finished and you know it all fits, you know it all works, then color over the pencil in pen. Okay? So graph in pencil until you know it's right. And then once you know it's right, then color over the pencil marks in ink pen. All right. Any questions for me on graphing? All right, so last time we started talking a bit about pedigree charts. And we know that pedigree charts show us lineages. They show us how traits are passed from generation to generation. And oftentimes from a pedigree chart, we can determine the mode of inheritance of a trait. How is it passed? We talked very briefly last time about several different modes of inheritance: autosomal dominant, autosomal recessive, X-linked dominant, X-linked recessive, Y-linked, and mitochondrial. Remember that an autosome is a non-sex chromosome. Doesn't have anything to do with your gender. Humans have 22 pairs, or 44 total autosomes in each diploid body cell, in each diploid somatic cell. We also have 2 sex chromosomes. And remember that in females, those sex chromosomes are pretty much the same. They're 2 X chromosomes. And then in males, we have an X and a little tiny Y chromosome. So when we're talking about gene linkage, genes are linked if they're found on the same chromosome. Oftentimes, linked genes are inherited together. Now, there are situations where things like crossing over can cause them to switch, but linked genes are genes on the same chromosome. So if we talk about a trait being Y-linked, that means the genes for that trait are on the Y chromosome. If we talked about it being X-linked, the genes are on the X chromosome. A carrier is an individual who has a copy of a recessive allele, but only one copy, and therefore they don't express the trait. They could pass the allele on to their offspring, but that individual themselves, they don't have the trait. They just have a single allele for the trait. Just a second, I'm trying to keep up with the pear deck as well. So we looked at some symbols for pedigree charts. Males are squares, females are circles. A colored in shape represents an individual that has a trait. A half colored in shape represents a carrier or an individual who's heterozygous. Sometimes instead of seeing half colored in, you'll see dots there. Oftentimes the dots refer to carriers for sex-linked traits. But what I want you to think about, whether it's half colored in, or it has a dot in the middle, those individuals have one allele for the trait, for a recessive trait, and they're not affected. Individuals connected through their centers by a horizontal line are mating, and then their children are connected by a vertical line and a horizontal line, like so. So this couple has 2 children, 1 daughter, 1 son. We can see dizygotic twins, so these would be fraternal twins, not identical, or monozygotic twins this way. Those are identical twins. And we want to be able to know how traits are passed on. We want to be able to determine the modes of inheritance of certain traits. So let's quickly run through the characteristics of these modes of inheritance. So traits that are autosomal dominant, this means that the genes for them, the alleles for them, are on an autosome, a non-sex chromosome. And dominant means you only have to have one allele to have the trait. So since these are autosomal, males and females are equally likely to be affected. These traits don't skip generations because there are no carriers. You either have it or you don't. When you have a gene, you have the trait. Males can pass these traits on to their male children. They can also pass them on to their female children. There's several human genetic conditions that are transmitted this way. Oftentimes we talk about disorders transmitted this way. Huntington's disease is a neurological syndrome. Achondroplasia is a type of dwarfism. This is a very common type of kidney disease, very serious kidney disease. And this one is a form of really high genetic cholesterol that can cause very early heart attacks and strokes. When we're looking at pedigrees for autosomal dominant traits, there's some things that we need to make sure we look at. So notice in generation number 1, this individual is a man with the trait. His mate, not affected. So these folks have some children, and notice they have a son who's affected in generation 2. They have a son who's not affected here, a daughter who's affected, a daughter who's not affected, and another daughter who is. Notice that this individual and this one and this one, these would be like their sons and daughters-in-law. Okay, they're the mates to their children. Notice that an individual in generation 1 was affected, several people in generation 2 are affected, and then the children of generation 2, several of them are affected as well. You also want to notice that affected females can pass it on to their daughters or their sons. Affected males can pass it on to their daughters. We don't see it here, but they also, yeah, they can pass it on to their sons. So either way works. All right, let's also talk about autosomal recessive traits. Again, the alleles for these are on a non-sex chromosome. This time, though, you've got to have 2 alleles, one from each parent, to actually have the trait. Because these are autosomal traits, males and females are equally likely to be affected. These traits can skip generations. That's because they're carriers. Some individuals can have an allele without having the trait. Only homozygous individuals have these. You got to have 2 alleles, like little a, for example. These traits can appear in siblings, brothers and sisters, without their parents actually being affected. Their parents could be carriers again. If a parent has a trait, though, the parent is actually affected, like little a, their children are going to, at a minimum, be carriers because they're going to inherit one of these little a's from the parent. Now, there's several human genetic disorders that are inherited this way. Tay-Sachs and PKU, pretty common, and also cystic fibrosis, a neurological syndrome. Here's a pedigree chart for a trait that's autosomal recessive. Notice that we start up here with a female who is a carrier. The male doesn't have it and isn't a carrier. Female eventually passes the trait on, the allele on, to this daughter, but not the son. Over here we have another family. The man's a carrier, his mate is not. They have a son here who's a carrier, 2 daughters who are not. Well, this son ends up marrying the daughter of the other couple. They're both carriers. Neither one of them have this syndrome, this trait, but because they're both carriers, they end up having a son and a daughter affected with the trait. But again, we see that the allele can be passed from father to son. It can be passed from mother to daughter. It can be passed from father to son, father to daughter, mother to son, mother to daughter. And that's because, again, it's an autosomal trait. And again, it's recessive, so you have to have 2 copies to actually be affected, like these 2 individuals right here. All right, now let's talk about sex-linked traits. So sex-linked traits are traits whose genes are on either the X or the Y chromosome. Sometimes instead of talking about them being sex-linked, we talk about them being either X-linked or Y-linked. So we get a little bit more specific. X-linked traits, no matter if they're dominant or recessive, are always expressed in males because us guys only have 1 X chromosome. So it doesn't matter if it's dominant or recessive. Whatever's on that chromosome, we express. And obviously females don't express Y-linked traits because females don't have a Y chromosome. So let's talk about X-linked dominant traits. So the genes for these are on the X chromosome. You only need 1 allele to express the trait. Turns out females actually are more likely to get these traits. And the reason for that is that they have 2 X chromosomes, 2 opportunities to inherit it. They could inherit it from dad, they could inherit it from mom. Either way works. So the trait's present whenever the gene is present. Again, that's because it's dominant. This one, there's no male to male transmission. And this means that fathers can't pass it to sons because fathers give a Y chromosome to their sons and not an X. Males get their X chromosome from mom. A female who has the trait can pass it on to the son or the daughter, but may not pass it on to either one because she has 2 X chromosomes. All daughters of a male who have a trait are going to inherit it. And that's because a male, his X chromosome is affected. And his daughter is always going to get an X from him. His sons are going to get a Y. So whatever's on this X chromosome, his daughter gets. There's several kinds of genetic disorders related to X-linked dominance. Hypertrichosis is when people have an excessive amount of body hair. Porphyria is a blood disease that was common in like the English royal families. We think that George III, who was king during the American Revolution, had it. So here's an X-linked dominant pedigree. Notice there's no skipping of generations since it's a dominant trait. In generation 1, we have an affected female. And notice that she passes it on to a daughter and to a son. Notice the son passes it on to every daughter. That's the way X-linked traits are always going to work if they're dominant. And notice that none of his sons get it. And that's because his sons got a Y chromosome, not an X. His X chromosome went to the 2 daughters. Notice this daughter in generation 3, she passed it to a son and a daughter, but not every child, because remember, she has 2 X chromosomes. Same thing in generation 4, this male passed it on to every daughter, but no sons. So that's an X-linked dominant pedigree. Finally, let's look at X-linked recessive pedigrees. So these traits are the genes for them, the alleles for them are on the X chromosome. They're recessive. So typically it's going to take 2 copies to express the trait, though, except in males, because remember, males have a single X chromosome, which means that these traits turn out to be far more common in males than females, because what's on our single X chromosome, as males, we express that. These traits are recessive, so they can skip generations. All daughters of a male who has the trait are either going to be affected or they're going to be carriers because they're going to get their father's X chromosome. Again, there's no male-to-male transmission. Fathers cannot pass this to their sons. And there's several common human genetic conditions that are passed this way. Hemophilia, common bleeding disorder, the most common form of color blindness, red-green. Also Duchenne muscular dystrophy, where you lose control of your muscles of your body. All forms of X-linked recessive traits. So here's an X-linked recessive pedigree. Notice we've got a male who is affected. That means this male might look something like this. And lots of times we'll use superscripts to indicate the genes on an X or Y chromosome. And notice his is recessive, so he's got it. Notice that all of his daughters end up being, she's not a daughter. All of his daughters end up being carriers, and his sons not affected because they got the Y. Let's look at this daughter right here. So she's a carrier. She passes an allele on to that daughter, and she passes an allele on to that son. That son is affected because his only X chromosome has this gene, which is the gene for whatever trait, in this case, red-green color blindness. Notice this daughter. She passes it on to a son who's affected. She also passes it on to a daughter who's a carrier. Notice this couple right here. This is unique. We have an affected male and a female who's a carrier. Turns out this combination is the only way that you're going to get affected females. This female is the only one in our pedigree who actually has red-green color blindness, and it is very rare. X-linked recessive traits, way more common in males than females. And again, it comes down to the fact that men have X and Y, females have X and X. All right, our last couple of pedigrees that we want to look at are Y-linked. Again, these only affect males because we're talking about genes on the Y chromosome. All sons of an affected male are affected. Females can't possess these traits. They also can't pass them on. There's a form of blindness, retinitis pigmentosum, that's commonly passed on this way. It's passed on from father to son, from son to son, to son, to son, to son. Every generation that's descended from that male, his male children and their male children and their male children's children's children are going to be affected because they're essentially getting the Y chromosome from the affected person unchanged. Here's what a Y-linked pedigree looks like. Notice that every affected person is a male and that affected men pass it on to every male child. So that one's easy to pick out. The last one we want to mention are mitochondrial inheritance. So remember, there is a little circular piece of DNA. It looks very much like bacterial DNA in the mitochondria. And that piece of DNA is essentially a small circular chromosome. And it has some important genes on it. Well, it turns out that children inherit their organelles from their mother. So your mitochondrial DNA comes from your mother. So whatever genes are on your mother's mitochondrial DNA, you end up getting that. So this is from a mother to a son or from a mother to a daughter. In this case, the gender doesn't matter. Every child of a mother with a mitochondrial trait gets that trait. If you have a copy of the gene, you have the trait because you only have one of those chromosomes. There's only one mitochondrial chromosome. So all the children of an affected male, of an affected female rather, express the trait. Males can express the trait, but they don't pass it on because your organelles are inherited from your mother. Most mitochondrial traits, as far as disorders go, affect cell respiration or your ability to make ATP. So disorders related to the mitochondrial chromosome can be really, really serious health problems. Here's what a mitochondrial pedigree might look like. Notice in generation number 1, we have an affected female. Both of her children are affected. Notice this male, he mates with someone who doesn't have it, and none of their children or their children's children get it. On the other hand, this female, every child she has gets it, and every child that her daughters have also get it. But notice that her son's children are okay because, again, they're inheriting their organelles and therefore their mitochondrial chromosomes from their mother. All right, let me get to the right spot on the pear deck. I'm going to ask you guys to do something here in a second. So first of all, in plants, same kind of thing works. Mitochondria have chromosomes, so do chloroplasts. And all the organelles, remember, are essentially inherited from the mother. So in plants, those chromosomes, the one in the mitochondria and the one in the chloroplast, are transmitted from the egg, the ovule, not from the pollen. So again, organelle DNA is maternally inherited, whether we're talking about in humans or whether we're talking about in plants, in the mitochondria or in the chloroplast. All right, so we've got several pedigrees here, and I want you guys to look at them and see if you can determine the mode of inheritance. So what I want you guys to do right now is I want you to look at pedigree A. I want you to go to the pear deck screen. And I want you up around pedigree A to write what you think the mode of inheritance is for the trait being shown in A. So I want you to tell me, is it autosomal dominant, autosomal recessive, X-linked dominant, X-linked recessive, Y-linked, or mitochondrial? So look at A, and somewhere around the picture on A, just write what you think the mode of inheritance is for that trait. And I'll tell you what, while you're doing it, go ahead and do A and B. A and B. Try to come up with the mode of inheritance for both A and B. Take about a minute to do that now. All right, so let's talk about this one. So the first thing you want to look at is does the trait skip generations? And the answer to that is no. There are affected individuals in generation number 1. There are affected individuals in generation 2. There are affected individuals in generation 3. So it doesn't skip generation. And also, are there carriers that are marked? And again, no carriers are marked. So if there's no carriers and the trait doesn't seem to be skipping generations, that should probably tell you that this trait is dominant. Then you want to know, is the trait X-linked, autosomal, Y-linked, and so on. So you look at, first of all, are males and females affected equally? So just count how many of each. So for females, we've got 1, 2, 3, 4, 5 affected females. And it looks like we also have 5 affected males. If males and females are affected equally, that's almost always autosomal. One thing you can check, though, is can both genders pass it to both genders of children? So here we see affected mother passing to son, passing to daughter. Here we see father passing to daughter. And then up here, we see this generation 1 father passes it to sons and daughters. So if both parents can pass it to both genders of children, that tells you that it's autosomal. So this is the correct mode of inheritance for A, autosomal dominant. Now let's look at B. So on this one, first of all, does it skip generations? Answer is no. There are affected individuals in all 3 generations. Are there marked carriers? Answer, no. So again, that tells us probably dominant. Then we look, are males and females affected equally? Are there the same number of affected males and females? So we count 1 male, 2 males, 3 males. So 3 affected males, and then 1, 2, 3, 4, 5 affected females. So females seem to be affected more often. If that's the case, more than likely this is an X-linked trait. We can verify by looking at who passes it to who. And what we see here is if we have an affected male, he passes it to every daughter, but no sons. And then affected females can pass it to sons or daughters. Well, that's a dead giveaway that this trait is an X-linked dominant trait. So let's go ahead and work through C and D together. So first of all, are there carriers or does it skip generations? And again, carriers, remember, can be marked with a dot or a half shading. So there are carriers. If there are carriers, that tells you the trait is recessive. Secondly, are males and females affected equally? And notice in Pedigree C, there are no females that have the trait, but there are several males that have it. So when that's the case, more than likely this is an X-linked recessive trait. Some of you might have jumped and put Y-linked on this because males have it and girls don't. But remember, females can't carry a Y-linked trait. They don't have a Y chromosome. So this has to be something on a sex chromosome. And the only sex chromosome that would work in this situation is the X. All right, going on down to the next one. Same thing. Does it skip generations? Here, notice generation 2 is skipped. Nobody in generation 2 has the trait. And are there carriers? Well, yeah, there's lots of carriers. All those individuals with dots are carriers. So if it skips generations or there are carriers, it's recessive. And then are males and females affected equally? Well, we've got 2 affected males, 2 affected females. So more than likely, if sexes are affected the same, it's an autosomal trait. And again, we can check to see who's passing it to who. Notice that this affected male gives alleles to sons. He also gives them to daughters. And this carrier female here can pass alleles to daughters or to sons. So if the allele can be passed from one parent to both genders of children, probably autosomal. All right, we've got 4 more of these. So I want you guys to try with Pear Deck. I want you guys to try F and G. So beside the picture for F and G, take about 2 minutes and try to determine the mode of inheritance on F and G. Go ahead and do that now, F and G. Before we get to F and G, I'm going to go ahead and look at E. So come back to your Zoom screen so you can see what I'm writing. So we look at E and we see that the trait doesn't skip generations and there are no marked carriers. So if it doesn't skip generations and there don't seem to be any carriers, that probably tells you that it's a dominant trait. And then the next thing you look at are males and females affected equally. Well, you see there's 2 affected males, but there's way more affected females. If that's the case, probably X-linked dominant. And again, you can verify by looking at who passes it to who. Notice that affected females pass it to sons and daughters, but only some of them. Affected males, like this one, pass it on to every daughter, but no sons. That's a dead giveaway that this is an X-linked dominant trait. Now let's look at F. So first of all, again, are there carriers? Yes, there's lots of them. And does the trait skip generations? Well, it skipped all of generation 1 and all of generation 2. So since there are carriers and it skips generations, this one's recessive. And finally, are males and females affected equally? Well, there's only one of each gender affected. Also notice that females are carriers, so are males. Remember that males can't carry sex-linked traits because they only have one X and one Y chromosome. So if there's a male carrier, pretty much always we're talking about an autosomal trait. So the mode of inheritance on F, autosomal recessive. Let's look at G now. Again, look for, does it skip generations? No. Do there seem to be any carriers? No. Since that's true, this is probably a dominant trait. And then you look, are males and females affected equally? And you count. And it looks like there's 1, 2, 3, 4 affected males. There's 4 affected females. When the traits are affected the same way, or the sexes are affected the same way, rather, usually autosomal. Verify by looking at who passes it to who. And notice that father can pass the son, mother can pass the son, mother can pass the daughter. We don't see it here, but more than likely, men can pass it to women as well. And that's a good idea that this is an autosomal dominant trait. Finally, let's get to H. Does it skip generations? Are there carriers? And the answer to both of those questions is yes. And when that's true, it's always recessive. Are males and females affected equally? And what you see is there's 3 affected males, no affected females. Almost always that's X-linked. And again, you might be tempted to mark Y-linked here, but notice that the females are carriers. And females cannot carry Y-linked traits because they don't have a Y chromosome. All right, so those are modes of inheritance. Are there questions that I can answer for anybody? Unmute and ask. I can't see the chat very well because of my screen arrangement. So if you have a question, unmute and ask it. All right, another thing that I want you to think about, every now and then they'll end up asking the question, what causes you to have the traits that you have? And I want you to think of you have the traits that you have for 2 reasons. One of them are genes, and the other one is environment. So your environment doesn't change your genes, but your environment can regulate your genes. The environment can determine when certain genes are expressed, when those genes actually make proteins. So genes plus the environment determine your phenotype, your appearance, both of them. And again, it's because the environment affects gene expression. Sometimes we might talk about what's called phenotypic plasticity. And this occurs when individuals with the same genotype, maybe like little a, have different phenotypes because they're in different environments. An example would be the color change of the Arctic fox and other like Arctic hares and such. So in the summer, they have this reddish brown pigment. But in winter, they become pure white. And it turns out that the genes that make the red-brown pigment, they're inhibited. The expression of those genes is blocked by cold temperatures. So the foxes stop making that red-brown pigment, and therefore their hairs become white. Another example, pretty common, a lot of you probably have this plant in the flower beds around your house, the hydrangea. So that plant, if it's grown in soils that are somewhat acidic, the flowers are this brilliant blue. But if they're grown in soils where it's a little bit alkaline, a little bit basic, their flowers are brilliant pink. So again, genotype affected by the environment, which in turn affects phenotype. Also, something to think about is that the drastic change in heights of humans. You know, the human height has increased dramatically over time. A lot of people think that has to do with our protein-heavy diet that we're eating now, opposed to diets that our ancestors ate. So if they ask you why an individual has the phenotype they have, genes plus the environment, both factors are really important. All right, we're going to end today's session by looking at a chi-square problem. First of all, I want you guys to take a minute and just read through the problem. And then I'll work through it and show you some things about it. So take about a minute and just read the whole prompt. All right, so let's go through and kind of draw out what we think is going on here. So this P generation, this is like generation 1. We have a female, so we'll show XX, who has white eyes. And she's crossed or bred with a male, XY, who has red eyes. So that's in generation 1. That's the P generation. In generation 2, their babies, all males end up with white eyes, and all the females end up with red eyes. And then in generation 3, so the F1 is generation 2, and the F2 is generation 3. Notice that in the F2, the 3rd generation, we get a mix of everything. First question, though, what is the most likely mode of inheritance for the eye color trait? Well, I'm hoping that you're seeing that the sexes are affected differently. We start off with white-eyed females and red-eyed males. We end up with white-eyed males and red-eyed females. So something's going on that's affecting the sexes. So more than likely, this is an X-linked trait. And again, we know it's not Y-linked because females are affected here. So let's think about some symbols here. This generation 1, P generation female, she's purebred for white eyes. We need to know is white dominant or recessive. Well, here's her babies. So her babies end up with red eyes. We know that one of her chromosomes, X chromosomes, ended up over there, and one of the fathers ended up there. She had X chromosomes with white eye genes. He had chromosomes with red eye alleles, and the babies ended up with red eyes. So I'm thinking that probably means red eyes are dominant. So let's do some symbols here. So if red eyes are dominant, let's put a big R on this X chromosome, and let's put little r's on these X chromosomes. Notice there's nothing on the Y, because these genes are X-linked. They have nothing to do with the Y. So let's draw a Punnett square that depicts the cross that produced the F1 flies. So the F1 flies are the children of these individuals. So if we draw a Punnett square, looks something like this. So these individuals would have a dominant and a recessive. They're going to have red eyes. They're red-eyed females. These are going to be white-eyed males. These are red-eyed females. And these are white-eyed males. So that explains what the problem told us we should get. All the females red, all the males white. So next it wants us to draw a Punnett square that depicts the cross that produced the F2 flies. So remember the F2 flies, we got a little bit of all the possibilities. We got red-eyed males, white-eyed females, and so on. So the F2s are the children of the F1s. Remember, the F1s look like this. Those were the red-eyed female mothers and the white-eyed female fathers. So if we draw a Punnett square between those, here's what we get. So some of the children should be red-eyed females. Some should be white-eyed females. Some should be white-eyed males. And some should be red-eyed males. Turns out 1/4 of each. 1/4 of each possibility. And we do want to break this up by gender because this is an X-linked trait. You always want to break those up when you're analyzing them. So it tells us in part D, out of 100 F2 offspring, how many were expected to be wild-type males? Well, we expected 1/4, which would be 25. We're expecting 25 of each of these possibilities. All right, and what we want to do now is compare what we expected to what we actually got when this breeding took place. And that's our null hypothesis. And remember, null means no or none. And your null hypothesis is always going to be there's no difference between your two groups. So in this case, I would say there is no statistically significant difference between my observed and expected data. That's a good standard way to write a null hypothesis. And then you're going to test to see if that's true or false. And we're going to do a chi-square test to do that. And we're going to do that on the next slide. Before we do, we need some numbers. So first of all, when you're doing a chi-square test, it's a good idea to do a table like this. And you're going to write in the first column your phenotypes. And notice my phenotypes. And then you've got each of these columns. So this O is observed, expected, observed minus expected, observed minus expected squared, observed minus expected squared over expected. So our observed numbers should be given to us in the problem. So let's go back to the prompt. We had 23 wild type females and 26 wild type males. Those are the red eyes. So 23 females, 26 males. So 23 of these, 26 of these, and then 25 white-eyed females and 26 white-eyed males. 25 here, 26 here. Check those numbers, make sure I got them in the right spot. 25 white-eyed females. Yes. So your expected numbers go in this next column. And the way to figure that out is add up this first column. And when you add that up, I think you'll see that adds up to be 100. Then you're going to take your ratios from your Punnett Square and multiply by this total population size, or total sample size. So we had 100 individuals. 1/4 we expected to be each one of these phenotypes. Remember, 1/4 of 100 is 25. So we expected 25 of each. Then we just do what the table tells us to do. O minus E. 26 minus 25. 26 minus 25. 23 minus 25. 25 minus 25. Then we go to the next column. O minus E squared. 1 squared. 1 squared. Negative 2 squared. 0 squared. And then finally, O minus E squared over E. So that would be this column divided by this column. You can go ahead and divide it out and do a decimal. I'm going to do a fraction because I don't have a decimal. I don't have a calculator in front of me right this second. So this would be 1 over 25, 1 over 25, and then 4 over 25, and then 0 over 25. And notice your formula for chi-square is up here at the top. So chi looks like a fancy X. So chi-square is the summation of O minus E squared over E. So that tells you add up this column. So your chi-square value is 6 over 25. 1 over 25 plus 1 over 25 plus 4 over 25. Let's calculate that. Let me get a calculator. So that comes out to be 0.24. Our chi-square value is 0.24. All right, by itself, this number doesn't tell us much. We have to compare it to a standard value from a chi-square table called the critical value. And the way you're going to do that is, first of all, you need to look at how many phenotypes or options you have in the first column. We have 4 options. Our number of degrees of freedom are always our number of phenotypes minus 1. So since we have 4 phenotypes, our degrees of freedom are 3. And it turns out, unless you're told differently, you're always going to use the p-value of 0.05. So you're going to look where these column and row intersect, and we end up with a critical value of 7.82. And then you compare these two numbers. So in this case, our chi-square value is less than our critical value. If that's the case, you're going to fail to reject your null hypothesis. And what that means is, yeah, there's some small differences between my observed and expected data, but those differences are small, and they're probably just chance differences. They're not statistically significant. Now, on the other hand, let's say that our chi-square value had been 24 instead of 0.24. Well, in that case, our chi-square value is greater than our critical value, much greater. And when that's true, you're going to reject your null hypothesis. And what that would mean is that there's large differences between our observed and expected numbers, and that the differences are probably too large to be due to chance. The differences are statistically significant. So we reject the hypothesis that there are no differences because there seem to be differences, important differences. All right, I think that's probably a good place to stop. Can I answer questions for any of you guys? All right, I want to thank you guys for being here. One thing that I really want to encourage you to do, the College Board on the AP YouTube channel has published a series of review videos for the exam. There's probably 3 or 4 of those for every unit. They're short, to the point. I would highly recommend that you watch those before you take the AP exam. We'll have one more of these sessions. It'll be next week, same time, same place, Monday, 4 o'clock, and we'll go through some last-minute exam tips. And we'll probably also talk a little bit about evolution and Hardy-Weinberg analysis on that day. All right, I want to thank you guys for being here. I'm going to check the chat and see if there's any questions there. Looks like we're good to go. All right, thank you guys for being here. Hope to see you next time.